Item hashtag, SCP-047. Object Class, Keter. Special Containment Procedures, SCP-047 is to be contained in a 0.5 mx 0.5 mx 1 m hermetically sealed storage box at all times. This box is to be locked in Storage Locker 047A, inside P3 Secure Biohazard Lab 047B. Any entrance to and activity inside 047B will be recorded by biometric scan, closed-circuit camera, and redacted. Entry to 047B requires the authorization of the project manager, in addition to at least one O5 level clearance. SCP-047 is to be treated as a Priority 4 contagious biohazard in all protocols, including mandatory quarantine if exposed. Suite Q-047 has been provided, adjunct to Lab 047B, for this purpose. In the event of outside contamination of SCP-047-1, Lockdown Protocol 047-01 Yersinia must be engaged. Description: SCP-047 is a heavily rusted, breached gas cylinder made of an iron-redacted alloy. When exposed to open air, the material of the cylinder evaporates slowly, producing a previously undocumented mutagenic gas. This gas has no effect on eukaryotic organisms, e.g. humans, but profoundly alters prokaryotes, showing preference for common human microbiota, the natural microorganisms that live on the skin and throughout the body. On rare occasions these mutations produce a superbug, collectively known as SCP-047-1, a natural commensal with enhanced survivability and therefore opportunistic pathogenicity. The pattern of changes induced by SCP-047 suggests that these highly infectious microbes are, at least to some degree, selected for. Although the specifics of SCP-047-1 species are dependent on the base bacterium from which it is derived, there are several characteristics which appear to be generally consistent across all cases of SCP-047-1 mutation. Enhanced survivability in the bacterium's natural environment and similar environments, full-spectrum antibiotic resistance, increased reproduction rate and consumption of available material, development of a sporulation ability in gram-positive bacteria, increased ability to uptake, hold, and share plasmids, particularly in gram-negative bacteria, increased transmission, due to traits described above. SCP-047-1 samples are normally debilitating and virulent. However, compared to other Keter-class SCPs, it should be noted that SCP-047-1 have a relatively low mortality rate due to their action through mundane biological pathways. Several strains of bacteria have been selectively mutated by SCP-047. Mutation of bacteria in culture is possible, but the process appears to be much more effective with bacteria living on a human host. Generally, mutation of natural commensals for experimental purposes is encouraged. After the containment breach of January 30, 2010, see Incident Report Yersinia 047-01, 2010, mutation of already pathogenic species is banned and all existing samples must be destroyed. 3 particular species of SCP-047-1 mutated bacteria are of note, due to their involvement in the containment breach of //201. SCP-047A.png. D15978 infected with P047A, 2 days after initial exposure. Propionibacterium 047A is a strain of Propionibacterium acnes mutated by SCP-047. Show details. Streptococcus 047C is a strain of Streptococcus mitis mutated by SCP-047. Show details. Clostridium 047A is a strain of Clostridium difficile mutated by SCP-047. Show details. Recovery Log 047. SCP-047 was recovered from the Site Secure Laboratory by a Foundation Biohazard Recovery Team in response to a full compromise situation on //199. Testing logs indicate that the research team was attempting to contain data expunged in a Class SCP stable pressure cylinder, which led to redacted combining with redacted. A full molecular biological analysis of this is available in redacted. The initial release of gas when SCP-047 was structurally compromised was sufficient to cause a microbial bloom of uncounted species of SCP-047-1, killing all staff in the lab within hours. Exposed site staff obeyed standard Foundation quarantine/containment protocol, and the infection was contained successfully.